Great. Well, thank you so much for being here today. I'm here with Dr. Judy Kim from Milwaukee, Dr. David Brown from Houston, and Dr. Charlie Wykoff from Houston. And first of all, congratulations on such an incredibly interesting Retina Day. You were one of the program chairs. I can't commend you highly enough about what a great job you did. But for you guys, what were some of your highlights over the past 2 days?
You know, I think some of the highlights was some of the new imaging technology, so much more OCTA from multiple different companies and venues, imaging that we've never had before, home OCT from Notal, that may really change the way we monitor patients, the way we do in the clinic. And then a lot of the new data such as brolucizumab coming out, a lot of big buzz for that, up and coming drugs that we have in the pipeline, from other anti-VEGFs to gene therapy to all kinds of different mechanisms.
Excellent.
I thought all those exciting drugs are great, but Dave Wilson's talk about the cost of these drugs coming up and the cost to our society is also thought-provoking as well. Speaking of the imaging devices, there are lots that are available in the market, but also that are being used in research, and it's able to detect changes in metabolic changes in the eye, and I think that can be also very interesting going forward.
Yeah, I think that's got a lot of potential moving forward as well.
Michelle, great to be here with you and everyone. You know, it really has been exciting. This has been a great subspecialty today. Kudos to the organizers, an amazing alignment of talks and discussion and imaging sessions, surgery, medical, everything. The thing that I find most impressive about this sub-day is sort of this drive towards durable treatment options. And there's a lot to talk about in that space, a lot of good shots on goal with durable anti-VEGF therapeutics coming along. The gene therapy stuff to me is particularly cool. I mean, Al McGuire and Jean Bennett gave this really sort of emotional and beautifully laid out discussion of how they sort of developed this field. I mean, in many ways, just incredible, their story and the images that they shared on the first day of subspecialty. And then where the field's gone from there has just really blossomed. I mean, there's now great data from multiple companies, two in particular moving into sort of this durable VEGF suppression with gene therapy with their Genx Bio and Adverum. Both of them showed, I think, outstanding data that's very promising. Both of them in phase 1 trials, so we need to get further along in the trial programs to, understand these safety and durability and efficacy signals more, but really promising.
So you touched on something just before about brolucizumab having gotten approval this past week. So what are your thoughts about how we now have a third, well, actually technically a fourth, approved anti-VEGF in the marketplace?
Sure, anything we have to give more drugs in our armamentarium to help patients, especially if we have drugs that may be able to extend the dosing regimen,
right.
Treat those treatment recalcitrant patients because you have such a high anti-VEGF blockade load. It's exciting. I mean, this is for macular degeneration. I personally think it's even more exciting for diseases such as retinal vascular occlusion and DME that are super high VEGF levels. So I think this is the opening salvo for brolucizumab, but a big week for them.
I agree.
Yeah, speaking of RVO, we had a couple of talks about LEVO trial from UK, as well as comparing with their SCORE study here, and that was really interesting as well.
Yeah, what did you think of the comparison on that?
Well, the study design was different, but first of all, it's interesting to know that anti-VEGF is very effective for these drugs, but both for these diseases. But both trials were about 2 years long, so it would be really good to have that SCORE 5-year data coming out as well.
It's great. The more approvals we have in this space, the better. I think it really is tremendous to be able to offer opportunities to patients. Every patient is an individual. Every set of pathology in the back of the eye needs to be considered in its unique circumstances, and it's great to have another option. I look forward to using that clinically. Many of us have been involved with these clinical trial programs, the Hawk and Harrier trial programs, for years at this point, so it's nice to see that sort of come to fruition, to have a commercially available product. And there's a lot more in the works from a research perspective with brolucizumab. We're still learning a lot about all the other exudative retinal diseases, so really exciting.
I agree. Dr. Brown, do you have anything to add about the LEVO?
No, I think RVO is the most VEGF sensitive disease. It's a pure play. It's interesting that We're getting more and more data, and because it's such a variable disease, a third of the population fixes himself, a third of the population is really severe with ongoing capillary non-perfusion, and about a third need a lot of injections but function pretty well. So.
Right. Yeah, speaking of capillary non-perfusion, I thought the talks, back-to-back talks about panorama studies showing that we can improve diabetic retinopathy severity score with anti-VEGF, but then we had another Ramin's talk afterwards saying, but the non-perfusion does not change. Right. They are still non-perfused. So it would be really interesting, going forward.
Yeah, I think longer term, I think we need longer term data on that before you guys can make a definitive statement.
And the OCTA is really going to help us, especially swept source OCTA that's faster. Certainly the Plex Elite, when you can do montages, you can almost replace a fluorescein angiogram. Not presented at this meeting, but debuting today in the hall, Optos wide field OCT came out, which really may change the game on us being able to get, once they can get registration in OCTA, it may be an incredible tool for seeing what's truly happening.
And also,
in that debate, are we really, are we improving capillary non-perfusion, or are we just making it look prettier?
And also compared to wide field FA, wide field OCTA will be helpful because it's not leaky, so we have less, things to worry about. Is that artifact or not?
Yeah, OCTA is very easy to interpret and very little artifacts in the diabetic retinal vasculature. It's much more complex, as Nadia Waheed and others have showed, when you have projection artifacts on deeper layers and you got multiple layers of tissue trying to figure out interdigitations of things we really don't know about. But for diabetes, it's much easier.
Well, on the other hand, though, diabetic retinopathy, neovascularization is easier to see with fluorescein angiogram than with OCTA,
Because it lights up like a Christmas tree.
Yes.
Doesn't it, though? Yes. I mean, every time you guys are up on the podium, you illustrate that again and again and again. So, yes. So, okay, so why don't we talk a little bit, we've talked about the anti-VEGFs, why don't we talk a little bit about some of the surgical presentations that were given today? I mean, there were a whole bunch of them, they were great.
Well, the thing that I really am interested in is macular hole surgeries back again.
Isn't that great?
It's a topic. Different ways to stuff it, and also there was a way to fill it in with amniotic membrane as well. So I think we're now able to treat some of these macular holes that are so large or refractory to surgery that couldn't be treated before, but now treatable.
Yeah, my biggest thing is the techniques. Macular hole is a very successful surgery, and so you're going for a very small area. But what you saw today was that the imaging for the videos is just getting so much better.
Aren't they?
Right, the camera capabilities, the processing, and we're able to really see more what the surgeon's seeing. And I think that's what excites people to come, because they can see a world-class surgeon, do amazing things and really see it. Bigger than Dallas is just like it happened.
Exactly, and learn from it and actually start doing this on Monday when they're doing their macular hole surgery or whichever other.
Maybe you don't want to be Carl Klaus at home, or Klaus Eckert, or be a little careful doing that in your hometown.
Yeah, well, yes, exactly. Great. Well, thank you guys so much for your time. Again, we're here with Dr. Judy Kim, Dr. David Brown, and Dr. Charles Wykoff, coming to you from Ophthalmology Times. Thank you so much.